Hey guys, welcome to another week. What we're going to be doing this week is getting into cells. So we're starting to get a little bit out of just the basics now. Pardon me, if you've had cells in biology, like in high school, this should be a relatively easy, chapter. It's more of a refresher, but of course, there's going to be some details in that are a little bit more in depth, as far as how cells work and things. So I think we should just get started. Okay, so. Um, I don't think it's any surprise, but all organisms are made of cells, right? The cell, as we had mentioned before, is the simplest collection of matter that can support life. You can't get smaller than cells and consider something to have or be alive, really. Um, the cell structure is correlated to cell function. So this is something we've talked about in, a whole bunch of different ways, like how molecules shape, determines their function. Well, cell structure is also correlated to function, right? Muscle cells are built to be muscle cells, and they have, particular structures in them that allow them to contract. Nerve cells are meant to be nerve cells, and they have a different structure. So we'll talk all about that when we get there. All cells are related by their descent from earlier cells. So now we're talking a little bit about how evolution has formed not only the cells that make up our body, which are pretty unique, but also some of the simplest cells in the world, which would be things like bacteria. These are all related to the original first cell. So that's what they're talking about as far as descent goes. So here's the outline, right? Cells are the key to life, right? We are, if you think of your body, it's a little bit of a mind bender. We are a sort of like colony of organisms, right? We are a whole bunch of living cells. Each one of them is alive, but they all sort of work together and allow us to be a whole living organism, right? So cells are kind of fundamental to understand. So we need to be able to see them, look at them, and work with them, right? So uh, how can we look at cells? We can use microscopes, so that's what we're going to start in, and you can see the rest of the outline. So cells are extremely small, right? Very, very, very tiny. Just to give you an idea, if you were to read, a page of text at 12-point font, right? The period at the end of the page, or at the end of a sentence, is about as big as the biggest cell humans make, and that would be a human egg cell, right? And only half of us make eggs, right? So they're crazy tiny. So we had to invent along the way different methods to sort of examine them to see what they were all about, right? So we use microscopes. Microscopes visualize cells because they bend light, basically, and make the image of a cell bigger so we can actually make out the details. So light microscopes are what we normally would use in a Rowan class, unfortunately we can't do that, but light microscopes basically use visible light, so just regular old, used to be sunlight, but now it's artificial light, right? And that goes through a specimen. So the specimen has to be extremely thin, right? So that the light passes through it. And then that light is refracted through glass lenses, which magnify the image, okay? So for a light microscope, which they're calling LM here, right? For a light microscope, the resolution is about 200 nanometers. Now, if you don't know what that is, a nanometer is a billionth of a meter, okay, or a millionth of a millimeter, if you think about it that way, or sorry, thousandth of a millimeter. So really, really super, super tiny, right? Bacteria are about 200 nanometers across. So light microscopes can see bacteria, but they really can't make out the details inside. Scale is really important to consider here because I think a lot of sort of understanding is lost when you don't appreciate scale, right? So what we've talked about so far at the very bottom of this picture here is we've talked about atoms, right, and molecules. We're talking about zero point 0.1 nanometers and smaller, right? So that's 1/10 of 1 billionth of a meter or smaller, right? Everything we've talked about so far, large biological molecules and all the other stuff, in the previous chapters, are at that scale. Okay. Um, Now, if you keep going up here, we're talking about really big molecules like proteins and lipids and things. That's 10 nanometers. And then we get into cell parts, ribosomes, we'll talk about in this chapter. Viruses, which are not technically alive unless they're in a living thing. Viruses are very strange. If you ever want to talk about that, let me know, but I don't want to get sidetracked in that conversation right now. Um, And then the smallest bacteria, you start to get up into there. Then, if you're talking about animal cells, like our cells, right, and plant cells, by the way, we're talking much bigger. And then we just keep going up in scale, and then we're at like the meter to 10-meter range, right? So, average sized human being, if you're a tall human being, a little over 6 feet, you'd be about 2 meters, right? So, look at the scale here and just look how far away, these light microscope specimens are away from people, but they're not the smallest things, right? So light microscopes are it really can't be used for anything smaller than about, 1 micrometer or a tenth of a micrometer wide. Okay, so um, techniques to enhance contrast. So cells, because they have to be because they are extremely tiny, and they also have to be very, very thin for us to see anything inside, really are pretty featureless, right? So if you look at the bright field unstained specimen up here, this is basically if you took a cell from your body. This is probably the cell from the inside of your cheek, so like the inner lining of your mouth, right? And you put it on a microscope, that's what you're going to see. Very, very little detail. Not the best way to study what's going on inside. If you stain it, though, and this has been stained with a compound called methylene blue, if you stain it, it actually can be much more contrast, right? So you can see the nucleus very well in there. You can see how the cell membrane is folded up around the outside, and little pleats in it, right? You can even see a little bit of structure on the inside, right? So much better. And then there are also other methods too. There's phase contrast. There's differential interference, which we also call Nomarski. Okay, so you can kind of see, almost looks a little bit more three-dimensional, highlighted little structures on the inside. Unfortunately, we don't have the capability to do that at RCSJ. Uh, Fluorescence, so this would basically be dyeing different components of the cell with fluorescent dye and then looking at the cells instead of under regular, room light, you would look at it under black light. And that would actually get the cell to glow, kind of like when you're standing in line at Creamy Acres, about to go in the fun house, right? So that would be fluorescence. That will highlight a lot of different structures that you won't normally be able to see. And then the most expensive ones, which are light microscopy anyway, would be what are called confocal. So if you've ever done anything with, any kind of imaging software, you know that you can take pictures in layers, right? So like, focus on the back of the specimen, then the middle, then the front, and then with a computer, you can basically stitch them together, and that makes a really nice clear image of what's going on in the cell from the top to the bottom, right? That's what confocal will do. Okay, so if you're interested in microscopy, hey, it's up to you. It's a really extremely useful skill to have. You can get hired, in most diagnostic labs if you have some microscope skills. You can go to this YouTube link right here. Okay. Um, Over and beyond what light microscopes can do, we can talk about electron microscopes. Now, I want to say electron microscopes are really not that common. They're extremely expensive. I used one when I was at Penn State for some work that I was doing, and $1 million plus price tag is generally what you're going to see for an electron microscope. So instead of you actually using light, visible light to image a specimen, electron microscopes use electrons, right? So they basically shoot a stream of electrons at a surface, whatever specimen you're trying to look at. The electrons will bounce off of the specimen, and then therefore, and a detector will form an image from that. So it's obviously very, very derived. It's not as easy as just, putting a glass lens in front of something and taking a look. Um, these electron microscopes have really opened our eyes, to a lot more, than we could ever see with, just our regular light microscopes, right? So scanning electron microscopy basically looks at the surface of a specimen, while transmission, or sometimes they call it tunneling electron microscopy, they can actually look through a specimen, right? So, you can cut a specimen and then look through. So really what you're looking at here in these two pictures is actually the same cell structure, but what you're looking at is the cut through version on the bottom, and you're just looking at the surface on the top, right? So we really can, break down living things, cell structures and that kind of stuff, pretty well now between light and electron microscopes, right? So I'm not going to quiz you or test you on this, but if you just wanted to see like a difference in, what electron versus light microscopes can do, Light microscopes are very convenient. They're relatively cheap. Almost every, high school bio or even middle school class, has microscopes in it. Electron microscopes and, extremely expensive. They actually take a whole room to house them usually. And, but the positives are that they can magnify a million times or more. So imagine what detail you could see by getting something a million times larger, right? So anyway, don't worry about all the crazy details and stuff here. I think it's just more interesting to look at the first comparison, right? The useful magnification for light microscopy is about 2,000 times, which is good, right? But it's nowhere near a million times, right? So electron microscopes are definitely the most powerful ones we have, and there are even other forms of microscopes that are used that are not in this presentation. One's called an atomic force microscope, is even more powerful than electron microscopes. So, we're really getting a different look at nature, by looking at it, so close up, right? This blew people's minds when they were first, think about it for your all of human history, you're just looking at a drop of pond water, as a drop of water. And then all of a sudden, somebody invents the microscope, you stick it under a couple of glass lenses, and there's this whole world of living things, little cells and stuff swimming around that you never knew existed. It was like, a complete shift in people's consciousness of, reality almost. So it was a really, really huge invention. Okay, so looking at cells is one thing, right? Seeing how the cells are actually arranged inside and, where things are, as far as proportions and arrangement towards each other. But what if we actually want to work with all these little pieces of cells to kind of investigate them more and, figure out how they work? You have to separate all the little parts of a cell. So imagine if I gave you a cell, a big cell, like like I said, a human egg cell or whatever. Could you actually pull the nucleus apart and the all the little cell components apart, the mitochondria and all the other things in there without damaging it so that you could study it? It's almost impossible, really, until we figured out that we could use centrifugal force to do the work for us. So cell fractionation, okay, basically fractionation means to break up a cell and separate the parts, is accomplished using a machine called a centrifuge. Okay, so a centrifuge, I like to basically describe this. Maybe this makes sense to you, maybe it won't. If you've ever gone to an amusement park, most amusement parks have these, or down, if you go down like on the boardwalk, in Ocean City or something like that, there's a ride called the Gravitron. Okay, basically you go into the ride, you lean up against the wall, right? It's a round, circular ride. And then all of a sudden, when the ride starts, it just starts to spin faster and faster and faster. And what that makes you do is literally stick to the walls, and eventually you'll slide up the walls, kind of like gravity doesn't exist, right? You're changing that force that's pulling you. This is how centrifuges work. They spin way faster than a gravitron, though. They spin to the point where we're talking about over 100 Gs, which means the force of gravity, the equivalent force of gravity would be like 100 times, right? Of what we would normally feel. What that does is it takes the denser parts of the cell, that have now been broken up because they've been fractionated. It takes the denser parts of the cell, pulls them down to the very, very bottom of the tube that the cells are in, and all the less dense, parts of the cell remain at the top. Okay, so it basically separates the cell components by density. So basically what you're seeing here is here's the cells in a blender, right? So you just, your whole cells, which are over here, you blend them up, right? It breaks up cell membranes and releases all the cell contents. So now you have what's called a homogenate, right? So in other words, this is an evenly mixed, sort of like cell milkshake, right? When you put this in a centrifuge, and this is a very, very simple example, you spin it super fast, and all the dense stuff goes down to the bottom of the tube, whereas all the less dense stuff goes to the top. Then you have everything separated, and you can do the studies. Okay, so that's going to end it for this video. We'll continue with the next one.